We're ready now for Robert.
All right, you should see my screen. Is that correct?
We do see it.
Excellent. All right, so in this session, I want to talk to you about, so we've seen a lot of stuff about, this is how we build models. We can do some stuff with the GUI. We can specify these things with mod files, but that seems like a lot of work, and it is. But we can avoid repeating ourselves and repeating things that have already been done by taking advantage of resources that exist out there that have for model sharing. And in particular, right now, I want to highlight two resources. First of all, ModelDB, which is a discovery tool for computational neuroscience models, and neuromorpho.org for neuron morphologies. So like I said, ModelDB is a discovery tool and repository for computational neuroscience models. You don't have to write mod files from scratch. You can go to ModelDB and you'll find literally thousands of them as part of published models that you can reuse or modify. You can go there to look for code examples. You can go there to try and figure out how somebody built a model. So many options, just having everything in one place. What do we have? There are, as of the other day, 789 neuron models. That's out of a total collection of 1,746 models. These are all published. These are all models that have published source codes that are published papers that are available. They are not just neuron models, obviously, because 789 is not equal to 1746. And in total, you'll find stuff there from 105 different simulators or programming languages. And it's useful to look at these, even if you're working in Neuron, to be able to see, what kinds of parameters or how do they structure their data? Even if they're not using the tools that you're most familiar with, you can still learn from looking at how other people approach the problem. There's models for approximately 200 different cell types, spanning across 21 or more different species, 62 ion channels, pumps, etc., types of ion channels and pumps. These cover a wide range of topics, ranging from sort of the clinically focused, so like, what about Alzheimer's or Parkinson's, to the more basic science questions like, how does spike timing dependent plasticity work? And the reason that we need to do this is because it's just generally not possible to describe everything within a paper. There's a lot of complexity to these models. Just imagine the morphology and describing all the details of the morphology. Well, there's a lot of complexity to this. And in practice, what we found is that basically a third of model publications, so a third of the model entries in ModelDB have 20 or more files that are that collectively describe the model. About a quarter of the files are over 5 kilobytes in size. And you just cannot do this, and you cannot capture everything in a paper. And even if you try to do this, then there's always a possibility that bugs and typos, errors, and omissions might distort the dynamics. And sorry, I apologize. Hey, Evan, can you help him with that?
Hey, how's it going?
Sorry, I apologize. So anyways, so there's a lot of complexity, and if you try to reproduce things on your own, or if somebody's, it's possible that when somebody describes things that they do describe, that there could be errors that are introduced, typos in the typesetting process, who knows? And so there's nothing better than just actually having the code available for this. The tricky thing, the caveat on this that applies to ModelDB, that applies to anything else that you might go out there and try to reuse somebody else's resources. The fact of the matter is, other people have other priorities than what you have, right? So they might be solving a different problem than you, right? Hopefully they are, otherwise, why are you doing it? So maybe the species is different, maybe the temperature is different, maybe the age is different. And of course, they may have made mistakes. So how do you decide this? How do you know if you can trust it? How do you know if you can reuse it, if it's applicable to your question? Well, there's nothing better to do than to go back and read the associated paper, right? Go look, compare the model and the results to other similar models. Don't just look at one that gives a specific result that you're interested in. We have tools both within Neuron and within ModelDB for exploring the model structure to help you identify, oh, well, this is how the A currents are distributed. I believe that, or I don't believe that. You can check the ion channels individually, and obviously, whenever in doubt, try to work with an experimentalist if you're not one, so that somebody's out there that can give you any sort of context that you may not have. So let's go. So here we are. So here's ModelDB. You can get there by modeldb.yale.edu. Takes you to this page. It's a nice landing page. We have a lot of information there. We do tweet out. ModelDB does tweet out anytime that a new publication is added to ModelDB. So if you're interested in trying to keep track of just what's happening within computational neuroscience literature, this is a great place to go and do this. Just to chime in on Ted's comment with regards to reusing code. Yes, generally speaking, whenever you're pulling from a resource, the typical custom is to cite the model authors, the original people. So like for ModelDB, it's a model author. For neuromorpho.org, it's whatever paper had the morphology, and then the resource that you also say, and I got this from ModelDB, and then here's a citation for ModelDB reference paper. And all of this information for ModelDB is how to cite ModelDB and suggested examples. But in any case, so what do you find when you go here? Well, you could browse by model name, but there is obviously quite a list of models. So in lieu of that, maybe you want to look for models that capture a given cell type. So if you can click on cell type and look at the list of model cell types or not. That's the problem with trying to do a live demo. You can look at different model concepts. So if you're looking for things, we've got things grouped together. What has to do with activity patterns? Well, there's bursting, oscillations, temporal pattern generation. We have all of these things organized here. You can look for different things. You can look for different pieces of metadata to describe your model. You can search for something. So let's say just down at the bottom, let's say that I'm looking for a disease thing. I'm looking for Alzheimer's. And so I can find, for example, I can find, for example, that there are 14 different Alzheimer's models that are available in ModelDB. I can click on one of them. Here we are. So this is what an individual model entry looks like in ModelDB. We've got a brief description, a link to the references, and then all the various metadata annotations. So this is a Alzheimer's model that was looking at a hippocampus CA1 pyramidal glutamatergic cell. It has certain ion channels that are associated with it. It was done in Neuron, and various other things that we might want to know. We can look. So this gives us a high-level overview of the thing. If I want to download it, I can just hit download zip file. I can look at this in the context of the modeling literature. Obviously, if you want to study things in the context of the entire scientific literature, you turn to PubMed or Google Scholar or someplace like that, Web of Science. But within the context of the modeling literature, we try to keep track of any time that a computational model cites a given paper. So I can see that there are these various models. So the model of Kuhlmann and Mayori 2012 cites this particular paper. So I can see how the different computational modeling literature goes together. We can look at graphical representations of the cell structure under the model views tag. So here it is. This is a model of a CA1 pyramidal cell. This is what their CA1 pyramidal cell looks like. You can mouse over it to get a sense of the distribution of ion channels. You can also click into this and explore very briefly. So here's, for example, the distribution of A-type potassium conductances within the cell. And you see here, for example, that the modelers for this paper, which includes Ted, the modelers for this paper chose a non-uniform value of A currents distributed across this cell. If you're interested in why, check out the paper. And then you can decide if you believe that or you don't believe this. But this gives you, if you think that's important, this is a thing that you really want to know to make sure that the models that you want to use have all of this. And you'll notice we've learned some things about the cell. We know what the cell looks like. We know how the channels are distributed. We know what temperature the model was run at. It's run at 35 degrees Celsius. This, by the way, is not Neuron's default. Neuron's default is for squid because that was what Hartley and Huxley looked at. They like it cold. So we know that the modelers here thought about temperature. And in fact, if I click into this, I see that there's 10 different mod file mechanisms that were potentially temperature dependent. And then finally, just in terms of the overview of ModelDB, we can go in and click, of course, on the individual files. So here we are. There's a README description typically for files. I can then go and look at, let's see. Oh, here is a calcium activated potassium channel. And so here again, this is all described using mod files. We see derivatives, we see functions, and you can get a reasonable sense out of what all of this means, even if you're not comfortable writing this yourself. This, by the way, is a tip that many people have found useful. Find something that's similar and see if you can use that as a launching point to modify it into whatever you need the dynamics to be, rather than to just doing it from scratch. So yeah, so this gives you a sense of what ModelDB is. I will point out that, again, this is all in context of the scientific literature, modeling literature. So you'll notice that many of these files have little asterisks next to them. If I click on this asterisk, it's going to point out that this particular calcium activated potassium channel, not calcium activated potassium channels in general, but this particular calcium activated channel has been used in a number of other model entries in ModelDB. So I can get a sense as to what the community has thought of this particular channel. Well, it's been used in CA1 pyramidal cells. It's been used in CA3 pyramidal cells. So maybe those are, if I trust those papers and I trust those authors, maybe those are places where I might want to use this model as well. If I want to do Purkinje cells, I don't see a Purkinje cell model in here. Maybe this is not an appropriate model to use within a Purkinje cell. So I can get some sort of a sense of context of all of that just straight from the website. So how do we actually do anything once we've found a model? So let me not use this one. Let me pick a different one. So here's a model, classic model of Manin and Sinowski from 1996. I can just go ahead and download that file. It's downloaded off screen because that's how this works. So let me, for the moment, stop that and just go. And I'm in my download folder. And now I can, blah, blah, blah. I now have a cells folder just because I have a cells folder, by the way, because when I look into this, the file was called cells. So here I am. I can look at the files that I have within my cells folder. Sure enough, I have a bunch of mod files. I have Hoke, which is another programming language in addition to Python that Neuron supports. And here we are. So I have this model. This is all the code for the Manin and Sinowski model. What can I do with it? First things first, I want to run the model. I need to compile my mod files. No matter what platform I'm on, Windows, Linux, Mac, I'm always perfectly happy typing enter an IV modal. Other platforms, you may have other options, but enter an IV modal will always work. So there we go. I've compiled it. So in theory, everything's ready to go. You'll notice that I didn't get any nasty red error messages. So I can launch Neuron, many ways that I can launch Neuron, but I like to do everything from Python. So I'll just type Python. From Neuron, import H and GUI. And then there we go. I have the Neuron main menu has appeared. Now that the Neuron main menu has appeared, well, I will now load my file. There are you'd have to look around. You'd have to read the README to see how you're actually supposed to load the file. But I happen to know, for the sake of this one, that the main file, the launching file, is a thing called mossinet.hoke. This is a weird name, but you'll find that a lot of historical models use this name just as a convention for reasons, as the entry point for their model. So if you ever see a mossinet.hoke or mossinet.py, it's almost certainly the main launching point for the model. So I want to load my model. So load file mossinet.hoke. I could have done this through the GUI, but I didn't, just because I didn't think of it, to be honest. So it pops up a nice little dialogue. This ties into a conversation that we had in the chat a little bit. You can use, Neuron provides this interface to allow you to specify whatever windows, whatever pattern. So I want this graph and this graph in the same windows, they move together. Okay, you can do that with a few lines of Python code. So here they put a couple of buttons together. Okay, so I'll say that I want to run the figure 1 demo. And one of the things that they looked at was the effect of different morphologies. So let's say that I have a layer 3 pyramidal cell. So I just click on the layer 3 pyramidal cell. And I can, let's say, zoom in out and zoom out. So this is the cell. If I wanted, I could rotate it. I could explore it in other ways. But this is the cell morphology. If I hit and then run, I see that it's generating some short bursts. And I did all of this, and I could look at a different cell morphology because, so here's a layer 5 pyramidal neuron. Again, we can zoom out on this guy, move it around, and so forth. So this is a different cell, and if I run this particular cell, then this generates bursts that have more spikes. And that meant something to them in the context of the paper. You can go back and explore what it means. But now you can imagine that you can look at this, you have the code, you can modify it, you can do your own experiments on it, and you can build on it. Just to show you one other thing, just because we won't otherwise, I showed you model view within ModelDB. Neuron also has a tool called model view. You get there from tools, model view. And it has a very similar interface, and you can click in and explore different distributions of channels and what's where within the cell that you have. So I know I threw a lot of stuff at you, but importantly, we explored ModelDB and we looked at downloading a model and running a model. Does anybody have any other questions on any of that? All right. If not, let's go back to the slides.
If you do publish something that used files that you got from ModelDB, please be sure to cite the authors of the work that made that code available, and also please be sure to cite ModelDB.
And if you ever forget how to do that, it's linked on the bottom of the page on every page on ModelDB, how to cite ModelDB.
Should be flashing in red with dancing chipmunks. Well, maybe not.
I'll add that as a GitHub issue.
Okay.
All right, so going back to our slides, let's see. Oh, I guess one other thing while I'm here, just to point out, you too can share your model on ModelDB, and I hope you do. Whenever you're ready to do so, you can just go to ModelDB's homepage and hit the big giant blue button, submit model, can't miss it. And you don't actually need a whole lot of information. The only thing that you strictly need is what's your code, can't share code without code, who you are so we can get in touch with you, and when you're ready to make it public, we need to know the citations. We do, however, provide a tool that I strongly encourage you to use for suggesting metadata. So in particular, if I have, if I have a abstract, so I've written a paper, right? All of these things, the presumption is that there's a paper. So I've written a paper and I have some abstract, and that kind of describes my model. I don't want to go through all the effort of maybe I don't want to go through all the effort of clicking and figuring out, well, okay, what did they call it? What's the standard terms? So let me just sort of go ahead and hit that big blue button. Let us find ModelDB keywords for you, and I'll just paste in my abstract. Not my abstract, it's actually Ted's abstract, but you get the idea. And then it's going to come back and it's going to suggest, hey, based on your abstract, it looks like we should annotate the following pieces of metadata. Does your model involve calcium dynamics? Does it involve A currents? Does it involve dendritic action potentials? And it so happens, in this case, every single one of these pieces of metadata does in fact describe that model from Ted's paper. But if it didn't, I could just unselect one of them before accepting the ones that remain selected. And just like that, it populates some structured metadata fields. Why do we do this? We tag things so that other people can find your code. Why do you want other people to be able to find your code? Then they'll build on it. They'll, as Ted pointed out, they'll cite you. This is a good thing. And this makes sure that science continues progressing.
And when it comes to promotions and tenure decisions, which many of you may be facing, or if you're not, if you stay in the field, you probably will at some point. The tenure and promotions committee is not going to be asking your buddies on the campus where you are. They'll be sending out requests to people outside your university and maybe people you don't even know. And if you've been sharing your code regularly and people have been reusing it and it's been influencing their thoughts and they've been able to advance their work because they can learn from what you've done, you're more likely to get good recommendations.
Fair enough. So we are also, as a side note, ModelDB is in the process of updating the website to be more mobile friendly, to integrate model view, and to really sort of encourage more meta-analysis across the modeling literature. So you can see where things are at by modeldb.science. It is my perpetual hope that this is coming soon, so hopefully this will be up soon. If you do have feedback for you, if you've used ModelDB before, or you've gone here and you found things that you like or things that you don't like, I do invite you to fill out a survey on ModelDB. The link is in the chat and also right here in the slides. Please do let us know how it's worked for you and what we can do to make it better. So thank you for that.
Oh, one last comment about ModelDB is, even if you're looking in ModelDB and you find somebody else's code, but it's not for Neuron, sometimes people writing in other languages, people writing with other programming for other simulation environments, sometimes you can learn a lot from their code. You can find out the names of the values of the parameters that they use that weren't necessarily clearly stated in their paper. So it can be helpful to examine source code that was done, say, even for MATLAB or just bare Python or anything else. So.
Very true. All right. So if there's no other questions on this, moving on to the other resource that I want to highlight right now, it's especially important for neuron simulation, specifically because of Neuron's emphasis and support for morphologically detailed cells, neuromorpho.org. Neuromorpho.org is a repository of neuron reconstructions. This is important because it's extremely tedious and time consuming to do all of the clicking that's done to zoom out and identify the shape of the cell, but there's no point doing a simulation that's includes a neuron morphology unless the morphology is realistic. So somebody has to go through the bother of actually making that reconstruction. And fortunately for you, many other people have, and moreover, they've shared their morphologies that they've reconstructed on neuromorpho.org. So they have over 181,000 reconstructions from 1,000 different cell types, spanning 400 brain regions. Again, so same sort of caveats apply for neuromorpho as apply for ModelDB. Not everything exists in your context, right? When you're using morphologies that you didn't reconstruct yourself, you know, keep in mind that they may have not needed it, right? Maybe they maybe they didn't care about the 3D shape. They only cared about a 2D projection, or they stained it in a way that got them the data that they needed, but it doesn't capture the whole cell, or the cell got amputated, or there's all kinds of sort of experimental artifacts that could shape the quality of the data and the applicability of it for simulations, right? One way of doing this, again, same thing with ModelDB. If you find something that's useful, and you're going to cite it, right, and neuromorpho, if you find something that's useful, be sure to read the paper, right? Why did they make this? Were they studying some disease? Because maybe that disease led to a different shape of the morphology. And if you're trying to study a healthy cell, well, then that's not going to work. Some things that you can try to do, you know, run your tests. Look for, don't just plug a morphology that you found on the internet into a simulation without actually looking at it, right? Look to make sure, because sometimes there are disconnected or barely connected sections. And you can do this by inserting a passive conductance, lower the actual resistivity and lead conductance, and then just sort of injecting a huge ton of current into the soma. If there's no pinch points, no places where current has difficulty flowing, then you'd expect the membrane potential to rise almost uniformly everywhere. Another thing that's relatively common, especially in older models, is what's known as z-axis drift and backlash. This is even, so basically literally, oh, it looks beautiful from this side, but then I rotate the cell and then I see that for whatever reason, the Z values are just horribly off. That's easy. You know how you look for it? You rotate the cell and you see if that's going. You should check the diameters, right? Are the diameters constant or do they vary? Sometimes you'll find morphologies where somebody didn't care about the diameters. Maybe they cared about the branching structure of the cells. Perfectly legitimate study to do, but not so useful if you're saying that morphology matters. So check the diameters. Do they vary? Maybe make a histogram. Look at that. So let's go and actually, let's go to neuromorpho.org. I have one conveniently handled link here. It's it's tinyurl.com/neuromorpho C91662. Here we are. I will put that link in the chat as well so that you can play along at home. So here we are in neuromorpho.org. We found a particular morphology that's of interest to us. We can see that they also have a ton of metadata. Key amongst this, of course, is the paper wherein the morphology came from. That would tell us if there's, well, you know, so I can tell from looking at the paper title alone that this cell came from a rat. If I'm not studying a rat, maybe I don't want to use this cell. So I have some various metrics that have been analyzed on the cell. I have metadata. So this cell came between days 33 and 57. It was, you know, stained using Lucifer yellow and so forth. I can go and every entry in neuromorpho has two different representations of the morphology. First is whatever the paper author provided. And so in this particular cell, we get this data, which I'm not going to explain, but this is what's known as a eutectic data format. And so what neuromorpho has done, in order to make everything comparable, they have standardized all of the morphologies. And there are pros and cons to standardizing, but it does at least give a consistent format. So here we are. I can click on a standardized morphology and it looks something like this. This is what's known as an SWC file. Fundamentally, the lines starting with a pound, just like in Python, are comments. And then fundamentally in each line, I have a point identifier, a type. So one indicates soma, an X, a Y, a Z. This is a diameter, I think, or maybe a radius, I never remember, and the parent point. So this is supposedly what makes this cell. Well, let's find out. Let's actually do it. So let's take that morphology file, and I will download this file and I will store it. I will store it in my downloads file folder. C91662.swc is what I called it. So let's try it. Let's see what happens. So here we are back in my terminal. And I am going to go back up to my downloads folder just because that's where I was running. And I will launch Python to get to Neuron. Again, don't have to do it this way, but I think it's good practice. From Neuron, import H, GUI. All right, so once again, my Neuron main menu has appeared. I can go to File. No, just kidding. I can go to Tools, Miscellaneous, Import 3D. That's how I import a 3D shape. And then I can say that I want to choose a file. So I'll just click this little button. And over here, then I get a prompt to select a file. And I have a ton of stuff in my thing. So I will just make my life easier and I'll filter for just the SWC files, which are still numerous. C91662.swc. I don't want to find it. Wait, why is it not there? Give me 1 second. Star SWC. Okay, I didn't save it where I thought I saved it. That's why it's not there. Let's try this again, shall we? This is the problem with trying to do things live. Download linked file as. Okay, it's in my downloads folder, C91662. Now I gave it an extra thing. So let's try this again. You see this too, right? Why is it not there? Let's try this one last time. Download linked file. There we go. What? I literally see it right there. Oh, because it put it. Thank you. All right, so it ended up putting a text extension at the end of it. So I will move C91662.swc.text to just be C91662.swc. That was an annoying thing that happened when I downloaded from Neuromorpho. So now you know to look for it. And thank you to the person on chat who figured it out before I did. All right, so fundamentally, here we are. We now have this file. So let's go back to Neuron. I will try it again now. Let's see. So now, C91662.swc, there it is. We successfully imported it. Well, we haven't actually imported it. If I did, if I were to try and make a shape plot within Neuron right now, I actually wouldn't see anything. So what I can do, however, so I'm looking at, this shows all the points. I can toggle that on and off. I can show the diameters. I can rotate this around. I can do all that sort of thing. What I can do, there's two things that I can do. I can either instantiate it directly, which I'll go ahead and do. So now in Neuron, I have instantiated this cell. So now if I go graph shape plot, sure enough, there's my cell. And I can click that box and hit 3D rotate, and I can spin the cell around. And as I do so, you'll see that there's no crazy z-axis error, so that's a good sign. I'm not jumping all around. You'd know it if it was happening. And so forth. The other thing, the last thing that I want to point out on this, I can also, instead of instantiating it, I can export it to what's known as a cell builder. And we also had this sort of a chat on the chat as well. You see a very stylized representation of the topology. Don't worry, it hasn't lost the shape. But in any case, I can use this to declare biophysics, ion channels that are expressed in various places. Maybe I put on everywhere, there's a Hodgkin-Huxley channel, let's say. And once I've done this, I can go to management and I can declare a cell type and I give it a name, okay? And then I'm done with that. I can export this to a file and well, I can save it fundamentally. And then I can load that back into Python and create a whole bunch of these cells, all with whatever biophysics and shape that I've loaded. Does that make sense? All right. And just for the record, of course, you can also, everything you can do through the graphical interface, you can do through Python. So just for the record, this is how you would load a SWC file through Python. And you can either instantiate it at the top level, which is what's done here, or instantiate it inside of an object. And that will allow you to make classes that have shapes. And then you can plot it either using Neurons Interviews graphics, the way that we've shown, or this is how you do a shape plot inside of Plotly. If you like Python's graphical interface, graphical libraries as well, you can do Plotly, Matplotlib, what have you, and we'll see some of that later on today. And with that, I think that's all I wanted to say about ModelDB and neuromorpho.org, unless anybody has any other questions. There's a question, there's a question that I think I'll answer on the chat. Just because I need to think about it. It's a little tangential.
Yeah, the question really depends on what do you mean by myelinated axon, and I mean, do you need it to be myelinated between branch points, or do you need to have myelin pieces at regular or irregular intervals? The general answer is probably the latter, because real axons can often have long runs that are not branched. And yet, if you were to stain for the myelin and look at it, you would see many, many nodes of Ranvier along the unbranched section. What you need to do in that case is to break the unbranched thing into sections, myelinated sections alternating with nodes of Ranvier. And that becomes a process of manual editing of the morphometric data, and it's more complicated. You need either actual information about how long the myelinated parts are, or you need to make a good guess based on fiber diameter. So.
All right, so Ted, do you want to take it up on networks?
Okay, let me see here. Let me go back to sharing my screen, desktop.